Welcome students. Today we are going to study about the antigens. It is one of very important topic in the immunology. Let's start with the antigen. Antigen. Any foreign substance when enter our body give response in the form of antigens. Antigen is a foreign substance. Antigen is a foreign substance. When enter in our body gives response in the form of antibodies, and these antibodies help to fight against the antigens. And the simple definition of an antigen is that antigen is a molecule that initiate the production of antibody and cause an immune response. And most antigens have a large molecular weight and are chemically composed of proteins, polysaccharides, but maybe also lipids and polypeptides and nucleic acids. And many other antigens have a large molecular weight and are chemically composed of proteins and polysaccharides. And these antigens are also called as the immunogens because these antigens evoke the immune response in our body. Therefore, they are called as the immunogens. The concept of antigen. Today in this video, I am going to explain about what is antigen, and I am going to explain the basic concept of the antigen. If you have a good knowledge about the antigen, you can easily understand the other cross-reactive reactions in the related to the antigen antibody interactions, and many other deep topics are related to the basic concept. So if you understand this basic concept, you can easily understand the whole immunology. So the concept of antigen, it arose from the fact that our body can distinguish between the foreign material and the self material. So our body easily differentiate the self cell and the non-self cell or the foreign cell. So antigen is mainly considered as a foreign particle, and when it enters in our body, it evokes the immune response. So in response of these antigen, the body induces the production of antibodies, and that acts against the antigen. Most antigens in humans are entered through many other ways, and we are also going to study how the antigen enters in our next videos. So let's see about the properties of antigen. The first and the very simple property, but you have to remember this property, is the foreign nature. That I say earlier that antigen is foreign in nature, and its degree of foreignness is measured, and this degree of foreignness decide the immunogenicity, because all antigen that induce the immune responses in the host are foreign to the body of the recipient, and the host body recognize the antigen to be different from the normal body components, and the immunogenicity of the antigen increase with the increase in the degree of foreignness. In the case of biological antigen, the foreignness increase with the increase in the phylogenetic dissimilarity. The more the dissimilar the antigen, the more it is foreign to our body. So the foreignness decide the immunogenicity of the antigen. However, there are some exceptions in that some proteins occur within the host might also induce an immune response, as in the case of autoantigen, but it is an exception case. The second property of the antigen is the chemical nature, and we all know that the most potent and commonly encountered antigen are made up of proteins and polysaccharides, but also they are made up of lipids and nucleic acids. But in the case of protein, the antigen should contain the immunogenic regions with at least 30% of amino acids like lysine, glutamine, arginine, glutamic acid, asparagine, and aspartic acid. And these are the main component amino acid which make a protein antigenic in nature. And the level of the immunogenicity also increase with the heterogeneity of the molecules, and the homogenic molecules are less immunogenic, but heterogenic molecules are more immunogenic in nature. Remember students, heterogenic molecules are more antigenic or more immunogenic because they are made up of two or more components, and these two or more components constitute a more potent antigen. Therefore, the heteromeric are more antigenic in nature and homomeric are less antigenic in nature. The third point is the molecular size. The molecular size, it has been studied that the antigen should have a minimum size of greater than 500 Dalton, and the molecular size of antigen is a crucial role in the immunogenicity of the molecule. However, low molecular weight antigens are attached with the heptanes, and heptanes make them the high molecular weight or large size carriers. And the low molecular weight substances are termed as heptanes that are considered as a partial antigens with at least one antigenic determinants. And this make the heptane, when heptane attached with a high molecular weight molecule, it become antigenic in nature, but as it is in a low molecular nature, so it is not immunogenic. Remember students, the size matter in the molecular size matter of the antigen. If it is small enough, it will not evoke a much immunogenic response, but its size is more than 500 Dalton, then it is sufficient to evoke the immunogenic response. The fourth property of the antigen is the molecular rigidity and complexity. In general, rigid molecules are good antigens as they can raise antibodies to a certain structure when compared to the less rigid ones. And it has been established that the rigid molecules are good antigens, but the complexity of the structure is also essential. Remember students, complexity of structure, because the more complex it will be more heterodimeric in nature, and this complexity of structure is an essential factor as the peptide antigen with a repeating unit of a single amino acid is less immunogenic than a molecule with two or more repeating unit of different amino acid. Remember student, a single antigen with the same repeating units of amino acid will less immunogenic, but if it has two or more amino acid repeating unit, it will more immunogenic, more antigenic in nature. So complexity plays a main role in the antigens. Let's see the antigen structure. The antigen structure. Now, the fifth point is the antigen determinant and cross reactivity. The antigen determinants and cross reactivity are the regions in the antigen molecule that is involved in the reaction with antibodies. And usually an antigen with two or more antigenic determinants can induce a good response. And the good response means the production of high amount of antibodies. Thus, a smaller antigen usually doesn't induce antibody production, as it is not possible for a small molecule to have a more than one antigenic determinant. If the antigen have more than one antigenic determinants, more antibody will bind. Like you can see in the picture, you study in the books that this is an antibody, and it has a antigen binding site which we call epitope, and the antibody binding site is the paratope. So the antigen, this is the antigen with more antigenic determinant, then more antibodies will bind it. More antibodies will bind to the antigenic determinants, and then when more antibodies bind with, there is more production of the antibodies. And usually the antigen with two or more determinant can induce the antibody production, but a single antigenic determinant cannot produce. So, but the cross reactivities also play a good role in the property of antibodies, because the cross reactivity of antigen is also an essential factor where antibodies induced by a different antigen and can interact with the another antigen. So remember, the cross reactivity is also plays an important role in the antigenic property. So students, I know you will remember good by good these things, that how many properties are there. 5 properties of the antigen is there. First is the foreign nature, that the degree of foreignness is measured. Second is the chemical nature, that it is mainly made up of protein and polysaccharide. Third is the molecular size, it should be more than 500 Dalton. Fourth is the molecular rigidity, the more rigid the antigen, it is more immunogenic. Fifth and the most important property is the antigenic determinants. More antigenic determinants, more immunogenic the antigen is. So you understand the 5 properties. So remember, if in exam time there may be question on this topic mainly. Uh, Students, let's see the last point, which is antigen structures. The molecular structure of an antigen is characterized by its ability to bind the antigen binding site of antibody. And antibodies differentiate between different antigens on the basis of the specific molecular structure present on the surface of antigen. And most antigens are proteins or polysaccharides, as I earlier told in my slides, that they may be capsules, flagella, toxin, fimbria of bacteria, viruses, or any other thing, and this secretion acts as antigens. But the structure of antigen might be different depending on the nature of the antigen, their size, and immunogenicity. And all immunogenic antigen have a specific structural component called as epitope or antigenic determinant. Another name of antigenic determinant is epitope, and the number of epitope differs in different antigens and determines the number of antibodies on a single antigen can bound to. And the structural component of interaction in antigen can be easily are different, and these determine the classes of the antibodies, how the antibody bound to it. We can study this in another videos. And the region of the antibody that interact with the antigen is called as paratope, and the antigenic determinant is called as epitope, and the region binding with the region on antibody that interact with antigen is paratope. So remember these two terms, epitope and paratope. So that's all with my lecture on the antigen. I hope you will understand. If you have any doubt or difficulty in this, you can easily contact me in the comment box, and I can solve your doubts, and I will review that doubts in the next video. So thank you for watching, and like and subscribe to my channel.